Assalamualaikum and hi everyone. Today's experiment is about inheritance, so please refer to the latest lab manual book. We are going to do two exercises. 5.1 Inheritance of genetic traits in human, and exercise 5.2 ABO blood group inheritance. First of all, let's see the learning outcomes for this experiment. 1. To determine the inheritance of genetic traits controlled by single genes in human. 2. To determine the inheritance of ABO blood groups. For the exercise 5.1, there are 6 characteristics that we have to observe. They are: shape of nose. Straight nose is dominant to curved nose. Earlobe. Free earlobe is dominant to attached earlobe. Tongue rolling. Ability of tongue rolling into U shape is dominant to inability of tongue rolling into U shape. Dimple. Individual with are genotypically dominant compared to those without dimple. Left-handed. The right-handed characteristic is dominant to left-handed. Hitchhiker thumb. The ability to bend thumb at 60-degree angle or more are genotypically recessive compared to normal thumb bending. Based on the characteristics, you have to fill in the Google form provided to get the class results and fill in the table 5.1. The link or the QR code for the Google form will be shared by your lecturer during the experiment. In the table 5.1, you have to calculate the observed frequency and expected frequency of each genotype in your class. According to the first Mendel's law, for the expected frequency, the dominant genotype is usually 3/4 of the whole number of the class, is equal to 0.75, and the recessive genotype is 1/4 equal to 0.25. But for the observed frequency, the amount of the dominant and the recessive genotype is calculated based on the class data from the Google form shared. For example, the observed frequency for the each phenotype, you get 25/30 equal to 0.833, whereas for the recessive, you will get 5/30 equal to 0.167.
So in this video, we are going to introduce the materials and apparatus that will be needed during the experiment. Okay, first we need 2 clean slides, right, and a lensing device. Okay, for the materials, we start off with the anti-A and anti-B serum, alcohol swab, sterilized lancets, some toothpicks, and a few labeling paper for the slides. Okay, for the first procedure, take 2 clean slides and put them on the tiles. Okay, take 2 label paper written with NTA and NTB. Okay, and paste them on the slide. For the next procedure, you have to clean both of your hands, wash them using the soap provided. Once you're done, let them dry. For next procedure, swing both of your hands for about 10 to 15 seconds and can prepare to prick your finger. Okay, next we are going to prepare our lancing device. Okay, so first take off the cap, put one of the sterilized lancet inside, twist the cap and remove it. Okay, put back the cap to the lancing device and set by twisting the setting to the maximum and pull the lever at the bottom. Okay, next you have to apply alcohol using alcohol swab to your middle finger. Take a clean one, wipe onto your middle finger. Take the lancing device, and press the button and use a new clean alcohol swab and clean the first drop. So now you have to place the next drop at the center of each of the slide. Slide 1 and slide 2. For the next procedure, you will drop NTA serum near the blood on slide 1. Near the blood on slide 1. Okay, and then NTB serum near the blood on slide 2. Now, use the sharp end of a toothpick and mix the blood with the serum. Make sure to use different toothpick for each slide. Okay, next you have to tilt the slide a few times to allow the serum to mix with the blood and leave it for a few seconds for the agglutination to happen. Make sure to do the same procedure for NTB slides. Next, to identify which blood group you belongs to, use the provided Eldon card pamphlet. Inside have information for you to compare the possible outcomes of each of the blood group. Okay. For this video, we wanted to show the example of the outcome. Okay, if you look at these two different slides, okay, so that is to show how different the appearance of the agglutination compared to the one that doesn't agglutinate. So this shows that the person belongs to B blood group as shown in this guideline.